diverse. They make up over a quarter of described species, and we think we know why they might be so diverse, and it's that they feed on plants. Now, I know that's a really surface explanation, but one hypothesis for why that connection exists is that host shifts themselves, so switching from one host plant to another host plant, can actually drive speciation. In my lab, we call this the host shift speciation hypothesis. I know it's a really original name, but go with us here. This is basically a special case of ecological speciation, where you have, in the process of adapting to these different host plants and this divergent selection going on, you develop reproductive isolation as a byproduct. So basically what happens is you have, come on slide, you have divergent selection, and with a lot of plant-feeding insects, they have a lot of fidelity to their host, so they stay on that host, they mate on that host, they just hang out on that host their whole life. You have a really strong impact on immigrant fitness. They don't do well if they switch plants. Hybrids perform really poorly on both parental hosts because they're adapted to this like non-existent intermediate environment. And then you have a reduction of gene flow for all of those reasons, and that could lead to speciation. So under this hypothesis, you would predict to see three things. First, you would think that you would see genetic divergence with populations clustering by their host plant. You would predict to see reproductive isolation, of course, between those populations on host plants. And you would expect that you would see ecological divergence, which is the thing that is driving that reproductive isolation. And this is really the key for this hypothesis. That host shift and that ecological divergence on those hosts causes reproductive isolation as a byproduct of adaptation. So I'll be focusing on these first two sections today, but we will have work coming out on the third shortly. So if the host shift speciation hypothesis really is happening, we would predict that we would see signs of incipient speciation through this method in populations and species of insects, that occur on a wide range of host plants. To that end, I've been working with the red-headed pine softline, Neodiptera lecontii, which as you can see is widely distributed and recorded on 14 different host plants. So if you've never heard of a softfly before, it's okay. I'll tell you a little bit about Neodiptera. It's a really great model system. It's very typical for insects, intimately associated with its hosts. All Neodiptera feed exclusively on the genus Pinus. Neodiptera lecontii is a little weird because it feeds on 14 of them versus 1 or 2 or 3 like the rest of the members of the group. And they're very closely associated with that host plant through their entire life cycle. They're experimentally tractable. I can go out in the field in the summertime and find literally hundreds of these colonies in the wild, and they're really easy to rear in the lab. We have little plastic boxes and mesh cages. You throw them on clipped pines, and you give them seedlings, and they're happy. And then finally, something that's a little unique for plant-feeding insects are these are really well studied. A lot of them are pest species, so obviously people don't want them to eat their pine trees in front of their houses or eat their Christmas tree plantations, so there's been considerable literature on these guys and how to control them. So I'll just go right ahead into what I did. This will be broadly broken up into two sections. First, I'll tell you about my genetic work. So I've done a broad-scale population sampling at the locations that are indicated in this map. It's 90 of Neodiptera lecontii, which is the species I showed you a picture of a second ago, and then one member of its sister species, Neodiptera pinetum, which is very ecologically different. It's limited to one plant that these guys really don't like. And I basically prepared reduced representation libraries using the DD-RAD sequencing method. That's published by Peterson et al. From the Orchestra Lab. If you want to know more about that method in detail, I'm happy to talk to you later, but it's a little too much to go into for the scope of this talk. And we basically used this to generate SNPs, which we called using 4 different methods. So this is a relatively new genome preparation method. So we weren't quite sure what the best method was. So we used 2 de novo methods, which basically means that we just took the reads and aligned them to themselves and called SNPs based on that. And then we used 2 methods that were taking advantage of a reference genome that's been prepared by another member in my lab that's giving a talk later, so stay here for talk. And basically we just filtered that out, so we had 1 SNP per one of these DD-RAD loci that we recovered, and we only worked with SNPs that were present in at least 70% of individuals, just for completeness sake. These are the numbers of SNPs we recovered, just for your information. Obviously it's a lot, so it's a really good data set to use. And then with these data sets, we did 2 methods of genetic structuring. First, we did admixture, which is basically just Bayesian clustering, which I'll show you an example of, so don't worry if you're not familiar with the method. And then a discriminant analysis of principal components, which again, I'll show you a picture of if you're not familiar with. So I'm not showing you the data. If you want to see it, I'm happy to show you. Basically, all of these methods give exactly the same answer, which is really cool. But just for sake of ease, we're going to be looking at the stack preference-based method, which recovered about 21,000 SNPs rather. So this is an admixture plot. If you've never seen something like this before, basically each one of these black bars indicates an individual. and it's colored based on what percentage of ancestry that this method thinks that that individual belongs to. So over several different replicates, we recover a K of 3, so 3 populations as being best for this. So this suggests that LeContier is actually 3 separate clusters or populations, and then they're colored based on how much that particular individual belongs to one of those 3 groups. So okay, there's 3, but what are these 3? So what we did was we actually looked at the location where each one of these individuals are collected, and you can see that it falls very nicely based on geographic lines. So we seem to have a group sort of in the Great Lakes region, we have a group in the Northeast United States and sort of the Midwest United States, and we have a group sort of in the Southeastern United States. Awesome. So this indicates that there's at least 3 genetic clusters corresponding broadly with geography within LeContier. Cool. So we went ahead and we just a priori, based on these results, clustered the individuals we collected into 3 different populations, 3 different clusters. We have the blue group, the yellow group, and the red group in the regions I described previously. So for the rest of the figures that you'll see, this is what the coloration indicates, which one of these regions they came from. So here's the DAPC. This is basically another method of calling population structure, where basically you do principal component analysis, and then you take multiples of those principal components and try to maximize the distance that's pulled between. So this is using, I think, 10 different principal components that explain about 80% of the variation. So as you can see, while it's not exactly perfect, not everybody that's red or yellow or blue is put together, this largely recovers the same thing, 3 clusters broadly based on geography. Okay, so this is also really cool because these different clusters that we've recovered with both of these methods independently also correspond to morphological differences we observe within the species. So the blue group up here has where is my pointer? Okay, blue group has extremely reduced pigmentation corresponding to the individual on the top of the page. They're actually missing at least 1 row of spots that's present in the other populations. The yellow group has 2 body morphs actually that aren't pulled out as being different in this particular analysis. There's a white morph body type that's prevalent in the mid-Atlantic region, and then sort of our classical LeContier that's fully pigmented yellow body, all pretty. And then in Florida, we actually have a different developmental morph where they have really dark pigmentation when they're little, and they retain a black head about halfway through the development, whereas all of the other populations are either born with a red head or lose a black head cap during their first larval instar. So this makes us feel really good. So this appears to be that there is basically geographic structuring. That's a little complicated, however, because each one of these geographic regions also corresponds to a very unique set of host plants. So it's a little difficult here for us to tell whether or not this is real geographic structuring or if it's geographic structuring that's then being complicated by ecological impacts of adapting to their host plants. So to explore that, what I did was I looked at one particular population where I had, one particular area where I had 3 populations that were sympatric, literally their trees are touching across 3 different host plants, and I'm going to share with you the results of that, some preliminary results of that analysis. Okay, so basically what we did was we collected lots of different colonies from each one of those trees in this area where they're basically touching each other from the 3 different hosts. And then we assessed reproductive isolation. First of all, we tracked adult emergence. So we wanted to see if there was any sort of temporal isolation between these populations. And we also conducted a number of no-choice mating trials, basically in little videos like this. And just right now, what I'm going to show you is whether or not they mated. And we're doing some more in-detail analyses in the future. And then we also tracked post-mating isolation. So if any of these females did mate, we tracked to see did they lay eggs, how did their offspring do, etc. And we're also in progress of doing some ecological divergence measures, including if there's divergent female opposition preference, so do they like one host over another, and we're testing larval performance on all of the different hosts. So today I'm just going to be sharing with you the reproductive isolation data, but keep your eyes open for the ecological divergence. So first I'd just like to show you some information. This is basically how many adults have emerged on a given day from each one of these populations, and although you can see that there's overlap between these, each one of these populations has a clearly distinct time when most of the adults are emerging. So this indicates that there's at least possible temporal isolation between these populations. So we haven't done statistics on this yet, but it's pretty clear that they're emerging at different times. So now I'm just showing you here what percentage of individuals mated from one of those populations. So they were shown in blue bars on the previous slide, so I'm continuing the blue just to help you remember. So you can see that females, if they mate with a male from their same population, or if they mate from males from one of the other 2 hosts, they really don't seem to care. They mate about half of the time. So it doesn't seem like there's any pre-mating isolation between these populations. Okay, But if you take a look at post-mating isolation, in terms of whether or not a female that's mated is willing to lay eggs, you notice that if she mates with someone from her own population, she'll lay eggs about half the time. But in the other two directions, she doesn't really want to lay eggs. It's about a half production. This is not statistically significant, but when you do look at how many eggs she does lay, if she does lay eggs, she leaves about 100 when she mates with her own kind. She lays about 90 if she mates with one of the other populations. But if she mates with a Virginia male, she lays on average 36 eggs. And this is actually inflated because one female laid about 150 eggs and the others laid 5. So that seems to indicate that there could really be some significant post-mating isolation in terms of the numbers of eggs laid between these 3 sympatric populations. Now remember, their trees are actually touching, but they seem to be isolated anyway. So this indicates that there could really be some role of ecology in structuring these populations. If we see isolation in these sympatric populations where there are, real barriers to actually cutting down gene flow, it's probably happening in populations where they're actually allopatrically separated too. So just to conclude, it seems like our broad scale structuring analyses that there are roles of geography and ecology in structuring the populations of Bacardi. And then again, looking at these sympatric populations, they also support the role of ecology in structuring populations here. So I'll be doing more detailed work of reproductive isolation, ecological divergence, and genetic divergence on both a local level and a broad scale sampling level in the future. So with that, I'd just like to thank all of these really awesome people in my lab manager, my lab mates, especially a whole bunch of really awesome undergrads who have helped me do egg counting and setting up mating assays and watching those videos. And then the UK NGS working group for just helping me figure out this DD-RAD stuff. And with that, I will take questions. Yes.
Right, I guess this is completely unrelated to your work, but do you have any idea why? You mentioned that northern group actually has lower coloration. So
yeah, they have produced melanin-driven pigmentation.
Yeah, and also you said that the southern group from Florida and stuff, they have that kind of black cap. Do you have any idea why there is that difference in coloration according to kind of southern northern range?
My guess is that it has something to do with their defense against parasitoids. So melanin in insects is often used as a way of immune almost. So a lot of times when like a parasitoid comes and lays an egg in something, the insect's going to try to defend itself by surrounding that egg with melanin. So it's entirely possible that populations in the south, just because it's a lot hotter, it's kind of more like tropical-ish. I know it's not tropical, but tropical-ish. They might have a broader suite of parasitoids that are actually exerting a really heavy predation pressure, parasitism pressure on them, and that might be a lot. It might not be a better investment to go, I'm going to go really strongly pigmented down here to fight off these parasitoids, but maybe in the north there's not so many parasitoids, so they are investing more heavily in something else.
All right. Just, again, one more thing. So can that be connected to the actual species of pines that they're living at?
It could be, yeah. Okay. So, this is literally like the beginning work with this group, so we don't have a total understanding of everything that's driving this, so we're right now going with the host really seems to be a good thing to start with, and then we'll investigate whether or not, it's something about the host, if it's a better, escape from predators or
all right, great, thanks.
Else. Yeah.
So did you look at just the genetics of this little subpopulation?
I didn't show it here. That little one, I have about 15 to 20 individuals that are included in there because I wanted to look at it more broadly. It looks like one of those hosts, which is one that I didn't show any more detailed analyses, the population from pitch pine, looks like it's pretty genetically differentiated from the others, but I only looked at like 3 or 4 families, so it's not enough for me to be like, yeah, they're really strongly differentiated. So, soon. Not quite yet. Okay, thank you very much.